I'm making tiny chips that can detect disease with light. Now these chips, just like computer chips, are made of silicon, which doesn't look transparent in the colors of light that we can see. But just like a red filter that only lets red light pass through it, silicon is also transparent, but to invisible infrared light. Silicon is a special material in that it has a very high refractive index. This means that when you inject light into it, it's very easy to keep it in there. This lets you keep the light in very tiny structures that guide it through lots of bends and turns, and lets you do lots of really cool things with it. The primary tiny structure that we use is called a waveguide. These structures allow you to direct the light through the chip, just like you would use a pipe to direct water through a building. In a waveguide, the light forms modes, or characteristic shapes, as it propagates. The most commonly used mode shape is called TE, and you can see images of it and the waveguide cross-sections in the middle column of my slide. Now, even though the TE light is mostly trapped inside the silicon, there is a small amount of it that travels outside of the waveguide, propagating through the air. My group is putting these waveguides into fluids containing disease markers, cells, or other types of bio-goo, so that this small part actually travels through the fluid. Our collaborators are designing special chemistries for the surface of the waveguides that will catch bio-goo of interest, and that little bit of light that travels through the fluid can see what is happening on the surface of the waveguide. I'm designing special structures that the light propagates through that can tell us by measuring the light after it leaves the chip if the goo of interest has been caught. The signal that I measure will shift based on how much goo is present, as well as how much light is outside of the waveguide to interact with it. This property could allow us to detect molecules that indicate disease, or detect bacteria or whole cells in our fluids. One particular goal that I'm working on is designing structures that could tell us not only if we caught the bio-goo, but also how big it is. For that standard TE light pattern that I showed you, you can only see a region very close to the waveguide surface, which, as an analogy, can't tell you whether you've caught a whale or a whole bunch of little fish. But by using a different TM light pattern, you can see farther away from the waveguide, and the whale will cause a much larger change in the measured signal than the fish. And by using both the TE and the TM signal, it's my hope that we'll be able to not only distinguish fish from whales, but also deduce the size of the whales. Now, in terms of bio-goo, that means that we'd be able to distinguish disease markers from whole cells, and we would get more information about the sample that we're testing. It's my hope that when complete, our tiny chips will allow us to better and more accurately diagnose diseases from complex, unprocessed samples like blood. Thank you very much.